I guess we'll get started. Can everybody in the back hear me? Yes, no? Yeah. Okay, great, wonderful. Hello, in case anybody doesn't know me, I'm Dr. Greg Barnes. I'm the leader of the NeuroReg / Developmental Neurotoxicology Research Interest Group here for the CIEHS grant. So we're happy to welcome today Dr. Robyn Tanguay from Oregon State University, and I'm going to introduce her. She is the University Distinguished Professor in the Department of Environmental and Molecular Toxicology, and she is also the director of the Superfund Research Program at OSU and director of the Sinhuber, right? Sinhuber Aquatic Research Laboratory. She received her BA in biology from Cal State University, San Bernardino, and she did her PhD in one of my favorite areas, biochemistry, from the University of California, Riverside, and did her postdoc training at the University of Wisconsin. Dr. Tanguay has really pioneered and championed the use of zebrafish as a model to be able to do systems toxicology. And so her talk today, as you can see here, is thinking outside of the tank to unravel and predict chemical activity mechanisms. She has published over 200 papers that I could find on PubMed, including 16 in 2022 and 4 already in 2023. So we're looking very forward to hearing her talk today and spending time with her in the research interest group and at dinner. So welcome, Dr. Tanguay.
Ha ha.
You have the medical spending card.
Good morning, everybody. Thank you for coming. Thank you for the invitation to speak. I'm going to give a little bit of a kind of a why we do what we do, the challenges that we're trying to address, and kind of the how we do it, and then a few vignettes of how we tackle the problem we chose to tackle. So this thinking outside the tank. So about 15 years ago, I had a challenge in my lab to come up with a lab logo, and it's in the right-hand corner. So this is really, it's not just a cute little phrase. It was really intentional. We're trying to use this model and try to advocate the use of this model to improve and protect human health. Yes, a fish to use that. It may be more acceptable, and we had a conversation this morning where this is feasibly done by using these models to translate. But let me tell you, 25 years ago when I started in this area, that was not even a possibility to use these non-mammalian models for human health. So it was an intentional kind of to drive this point home every time I give a talk. So the outlines, I'm going to talk about what I find it, decide what are the challenges that I see in environmental health sciences and what I'm trying to address, at least for the talk today. I'm going to give examples of how we're using the zebrafish to fill important knowledge gaps that many stakeholders have. And then I'm probably going to do 2 of these examples of PHs, which is the center of our Superfund research program. PFASs, forever chemicals. I'm sure you all are hearing about those all the time. I'm showing how we're throwing our abilities into that space. And then I'll talk a little bit about, if I have time, some of the adult consequences from developmental exposures. I can't help but when I see a problem that I think we can address, we will invest heavily in it, and we try to advance that field in that area. One thing I'm going to talk about for some of the students in the room, I know there's a lot of students who are not choosing to go to academia. Believe me, my students are running to the hills. But I really want to say that, I mean, there isn't a better job, and Chris would probably agree, there is not a better job. If you can succeed in this field, there's nothing better than getting up in the morning and knowing what you do is important, and you can't wait to get up. I mean, that to get to work. And as long as I've been in this field, I still feel that way. So I hope to convey some of that enthusiasm today. So what do I think are the challenges, the big challenges in environmental health sciences? We need to make informed chemical decisions a lot faster. So one thing that, I'm not a trained toxicologist, right? I've directed 2 training grants in toxicology. I've built 2 training programs in toxicology. I've never taken a toxicology class. Don't tell anybody. So I'm disappointed in the field, right? If you look at the number of diseases that we now know are associated with environmental exposures, and if you did a PubMed search in the field of toxicology, how many toxicants are being studied for the association of those diseases in great detail, it's a really small number. And if you look at what the EPA is challenged with is, there's 80,000, 100,000 individual chemicals, and then their transformation project and their mixtures. We're talking about infinite combinations of chemical exposures. Most of them probably are not adversely affecting human health. We got them, but some are. How are we going to discover them? Are we going to discover them with the approaches we've used in toxicology for 50 years? The answer is no. So we want to say, how could we jump in and help solve that problem? And I want to be more forward thinking. We talk about these challenges, all these chemicals in our daily lives. We're making new ones every year, right? How do we jump in proactively to identify the chemical safety or biocompatibility earlier in the game, and then give that information to people who can make a decision before they deploy the chemical in commerce? We work, I can't even give examples because I'm not allowed to. We work really heavily in this space. We work with companies that are tasked to make high performing products. That's what their company does, but they'd like to make them safer. Interestingly, there's no federal mandate to make them safer. They're not, unless it's a drug or a pesticide. I'm surprised at how many companies, once they gain your trust, they want to work with you and say, hey, could you give me 100 compounds? Which ones are better? And then, so we've been doing a lot of that work. So that's funding a lot of my lab. But what I really want to know in a big picture is what chemicals at relative doses have the intrinsic ability to interact with biology to produce an adverse outcome. See, there's predictive idea there. Not all chemicals can produce an adverse effect. The ones that do absolutely must be interacting with something that's important biologically, or they wouldn't be active. All of that information is baked in the structure and the composition of the molecule. So we should be able to crack that code. It's not easy. It's not infinite, but it's not impossible. So then once you do that, if you could quickly identify the chemicals that are active and inactive, then you could start playing fun games with chemoinformatics and machine learning to figure out what are the structural features that are commonly associated with safety or commonly associated with adversity. So then how do we then mechanistic work, which I'm going to give a couple examples today. How do the chemicals that are active, how are they active? What pathways, for example, do they perturb? And then finally, how do we take this entire field and make it more predictive, which is what the many goals out there. So there's lots of ways to approach this. So if you work for National Comp Tox Program or Tox 21 Consortium, you try to create a bunch of assays that you think recapitulate some aspects of complex biology. And then you build these in vitro systems and you can make them high throughput. So you could look at reporter assays or in this case, reporter lines, look at cell death assays, proliferation, nuclear receptors. This can become a really big list. So when you new approach methodologies that I'm sure you're all hearing about, it's this concept that you can take and build a bunch of these in vitro platforms that in their totality recapitulate the complexity of biology. Yeah, it is naive, but that's kind of where the field is going. We don't want to do that because I think it's not tenable. I think it's going to be impossible to comprehensively create in vitro panels that can predict molecular responses, and let alone cell-cell interactions and organ-organ interaction. So we don't do that. And then finally, if you get a hit in these in vitro assays with a chemical across the concentration, is it adverse in vivo? If you're a decision maker, are you going to make a decision based on a blip in a plate reader assay? It's a challenge, right? So we said we go in vivo, and then we'll swing back to some of these as confirmatory for our assays. So what we wanted to do is we wanted to see if we could tackle big pieces of chemical space, get as much chemical diversity as we possibly can, and bring it to our system and be able to separate the chemicals that were active in this system versus the inactive. So you have to have some confidence that the system you're throwing it at is responsive to lots of different modes of action. So this is where, when I talk to my drug discovery folks, is drug discovery is a piece of cake relative to toxicology, right? Say you're doing a rational drug design. You have a chemical target. You're trying to create chemicals that modulate its activity in a precise way to precise concentration, hoping that there's no off-target effects. That's a really small box you're shooting at. Toxicology, you have a chemical, you have absolutely no idea what it does. Trying to solve that riddle is a little more complicated, and trying to do that for hundreds of thousands of compounds, it gets a whole lot more complicated. And yeah, that's an aside. So what we did, so we've convinced ourselves over the last 30 years almost that early life stages are the most susceptible period of time for chemicals. And this is the best point to use the data here to separate the actives from the inactives. So the idea is, and this is for the developmental biologists in the room and developmental neurotoxicology, et cetera, we know that early life stages are more susceptible to chemical exposures. And we know in theory why that's the case, right? This is the most dynamic period of an organism's life. You know, you're going from single cell to this complex organisms. The fate of cells are being determined, organs are being formed. It's also where, and everyone knows, it's the most conserved life period. So if you're going to try to translate data between a fish and a human, the absolute best time to do it is during development, because that's where the mechanisms are most common. And most importantly, if you actually, and this has been done on a few cases, if you would just track the expressed genome in fish or even in mice or in humans, almost the entire gene is used to accomplish a developmental event, maybe for a short period of time in cell specification, for example, proliferation. So these gene products are being turned on and off across this developmental span, so much so that every gene is touched upon to solve it. So if you throw your chemical when all the gene products are present, the probability of detecting an interaction between your chemical and a gene product that you don't know what it is during development. And then if those gene products are important, it'll perturb some event in development. And then the animal will report back to you that, oh yeah, that exposure did something. So this is why we use this as kind of like the ultimate biosensor of chemical activity. It's not gratifying for some people, that resolution, but it's incredibly valuable to have that ability to be able to detect a chemical that produces an adverse effect. So basically, we're just being able, we throw these guys in individual wells, and we just want to be able to detect adversity. So the more things you measure, the more sensitivity you have detecting adversity or activity of your chemical. So, and for a number of animal care and use protocols, we need to stop most of our high throughput screening at 5 days or 120 hours post fertilization. So across this timeline, this is how we do currently how we do the high throughput screening. So we start early on. For a number of reasons, we decoordinate our embryos at 4 hours post fertilization in mass. So we do about probably 20 to 50,000 a day. And so we can produce limitless quantities of embryos now using these instruments that we built for housing massive amounts of animals. So that's not a problem. We are not limited there at all. And then once they're decoordinated, they're really fragile. So, and these are 4 hours, they're really tiny. So if they touch air, they blow up. They touch plastic, they blow up. So you have to create, and so we have these robots that we built that pick them up and singulate them and put them into multi-well plates. And then we've demonstrated over the years that there's all kinds of problems when you do concentration response exposures. Low concentrations, chemical absorption wipes out your low level exposures. So you have these concentration response curves that go nothing, 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 and you have these steep curves. Oh, maybe it hits, you saturated a receptor, or maybe the chemical didn't hit the animal because you're plastic. So this avoids that by doing direct digital dispensing. Because we can do this in vivo and high throughput, we do massive replication. We kind of had to with our power analysis. So we'll do like 32 replicate animals at every concentration, 11 concentrations for every chemical, right? So the math adds up really fast. Our first measured assay, we developed this instrument for embryonic photomotor response assay. So it's basically folks who work with zebrafish when the spontaneous motion that kicks in around 18 hours post-fertilization. We built an instrument that can detect that. And it basically, there's a camera down here, there's an infrared grid up here, and the plate goes right there. So we can infer the motion using pixel changes. And then when we blast these animals with bright light, it stimulates this activity. And that's why these bars go up, the peaks go up, and then it goes back down. Pretty boring assay, but it's a beautiful assay. There's no moving parts. You're doing high throughput screening. You don't want anything moving because everything breaks when it moves. So this is a great assay. This is a really sensitive assay. It detects chemicals that are going to produce some adverse effect later in life. We detect a hit with this assay. It does not mean this is a neurotox assay. It often is, but it's not always. So this is just a biological readout of the chemical did something to the system to affect its ability to respond to this pulse of light. And then we do nothing until the end of the 5 days. Then we take it into another room where you have a whole room full of these units that detect motor activity. So basically, when the animals are in the light, bright light, they tend not to move. Turn the lights off. There's lots of activity, so you can turn the light switch on and off and measure their motor responses over time. And again, we do this for every single animal and every replication. So we have software that can pull all that data out now. We collect that, and then we also do our startle response, larval startle response, where we keep the lights off, and then we acoustically startle them to see whether or not they respond. And then we take it to another room, and we euthanize the fish, and then we look for features. We try to see anything that's adverse. And granted, these are really, quote, boring endpoints, but they're not. They're really, really common endpoints, but they're not nonspecific. So, for example, I wrote my first book chapter when I was a postdoc, and picture up here is on the cover of the book that this is a characteristic signs of TCD toxicity, because that was the only chemical that we and anyone in the world was playing with at the time. So every time we expose zebrafish with dioxin, they look like this. We've now done hundreds of thousands of chemical exposures, and this is characteristic for teratogenic chemicals. So lots of different modes of action produce these same endpoints. From a screening point of view, that's great. You can harness that and channel all your detection to detect these endpoints, and you're able to detect activities from lots and lots of modes of action. So that's kind of a principle of that. All right, so now first example. So polycyclic aromatic hydrocarbons, really common in the environment. We produce them through certainly oil production, but also forest fires, et cetera. In addition to these thousands of pHs, they're environmentally dynamic. So they get atmospheric conditions, they get hydroxylated, nitrated, oxygenated. And if you know anything about structure-response relationship, that creates a huge nightmare. Not all of these pHs, it's impossible that they can have the same mode of action. And humans are exposed to these in mixtures, and the mixtures vary based on the sources of exposure. So how do you tackle that challenge? We know that humans are mostly exposure from ingestion or inhalation, depending on the situation. So I started my career in toxicology by studying the H receptor pathway. We know that if you do a grab sample, there could be hundreds of different pHs in the environment. And some of the parent pHs, that means are just fused rings and no substitutions, or they can have various substitutions at various carbons on the rings. Interestingly, the EPA has been tracking maybe 24 of these things for carcinogenicity. Just a couple dozen of the thousands that are out there. It's amazing, actually. And they make some assumptions about the activities of the other ones. We know that some of these pHs interact strongly with the H receptor and activate the aryl hydrocarbon receptor pathway. We couldn't predict which of these pHs could do this, but I think we're in a pretty good position to do that. But that's not enough. And I'll show an example today is a weakness of these new approach methodologies. So the idea is that if you create an assay for the H receptor and you throw a chemical on it and it's a hit, then you're going to try to make some risk assessment decision based on that activity of an assay in that assay. It makes all kinds of assumptions that the other things that chemical does are similar. So, for example, you can have a compound that activates the H receptor. You say induction of this reporter gene that induces CYP1A as an example. That chemical may not be toxic at all. Oftentimes it's not. It's an adaptive response. It's metabolized, it goes away. That's one scenario. Another example, the chemical hits, activates that receptor, and the other gene expression changes that are induced are different. They're not the same as dioxin. And CYP1A isn't the important gene. It's all the other genes that are being modulated. So there's all kinds of trappings in these high throughput reporter-based assays, making assumptions on what that reporter is telling you. And our in vivo data is a cautionary tale to those folks. So we really right now can't look at a pH and say, we think this is going to be toxic or not toxic and carcinogenic, developmental toxic, but I think we're getting close to being able to do that. So proof of concept. So Kim Anderson, she runs a chemical mixtures core in our Superfund program. So she's an outstanding analytical chemist. So she was able to acquire as many bona fide pH standards as possible, gave them to us, put them in our DMSO as a solvent. And they're either the parent pHs, kind of this purple compound, or very substitution, methylated, hydroxylated. This is a really complicated slide, but there's just, let's say 126, I think, pHs in this. And we said, blind us to each one of these compounds. We're going to run them side by side in our system. So we ran it through what I described to you. So we're measuring things such as all these malformations. There was, at the time that we did this, there was 22 individual features that we can measure. We can now measure them with machine learning. We had this early behavioral assay. We had this 5-day behavioral assay. And in addition, because of the potential role of the H receptor, we have a reporter gene now, a reporter fish that measures CYP1A induction, GFP, CYP1A promoter driving GFP expression. In this case, we used immunohistochemistry. So we're looking at the whether CYP1A was induced, the protein, and then where it was expressed. And this is right out of the gate. It's like, this is interesting. So we had some compounds that we don't see any induction. This is a little bit of yolk. We have some where the CYP is only induced in the liver. See some where it's mostly the skin and the neuromass are expressing it. And then we have some where the vasculature, this is really what it looks like if you expose the fish with dioxin. And then some which is more the gut, and then some are just the skin. So in addition to being able to profile the activity of every pH based on whether it produced a morphological phenotype or behavioral phenotype, and at what concentration, we can then say, was CYP induced, and then where was it induced? So there's probably about 600,000 animals that were used to do this study here. And what we tried to do is try to cluster them. So you can't read the chemicals are over here. These are all the various endpoints. We have endpoints that are kind of got shifted morphology based. We got the embryonic photomotor response, larval photomotor response, and then CYP. So wherever you see a no color, that means we saw nothing. So this is reporting the lowest effect level effects. So some of these, and then we clustered in. So in category 8, 7, 6, 5, 4, we see no morphological effects at all, even though the concentrations were run at pretty high concentration. Category 8, we saw nothing, no effects on behavior, no effects on any developmental processes. And then this category 7, we only see effects in the larval photomotor response. Every single of these compounds affects the LPR, but nothing else. And then various combinations. This group up here, this group 1, these are the ones that are really active. So where you see the dark blue, where you see the dark blue means that happened at a very, very low concentration. These are really active molecules. And the yellow means you took a higher concentration to see effects. So most of these chemicals were not active. So we could sort them. So the idea from a mixtures point of view, remember I told you these chemicals never occur by themselves. So we have hypotheses that these 6 chemicals behave exactly the same way, likely have the same mode of action. So if we did mixture studies, which we're doing, these will probably be additive. We can predict what will happen based on the relative contribution. And similarly for each one of the categories. So we're working our way through those mixture stuff. But now what I think we can do, if we can explain the mechanism by which these chemicals affect this photomotor response assay, we could just pick one, explore it in detail, explain its mechanism, do it for another category, explain its mechanism. Before you know it, we might be able to explain the mechanism of every single pH just based on the activities that we can identify in like 5 days. So that's kind of where we're heading to. All right, it's problem. So I talked about all the merits of the great endpoints are sensitive to everything. They commonly occur, easily detected. We built instruments to screen them, very reproducible, and are responsive to lots of toxins. Huge disadvantage, they're kind of blunt readouts of activity. They're not very gratifying. It's like, well, every chemical does the same thing. They rarely, I would say, they never provide mechanistic insight. That's okay at this point because we're trying to use the biology to screen chemicals. We're trying to learn something about chemistry more so than biology because that's the part of the field I'm trying to address. I could study one chemical for 50 years, or I could tackle like all of the exposome in my lifetime, right? So I'd rather do the latter. Different initiating events. If you're an adverse outcome pathway person, different initiating events can produce the same endpoints. And we've shown that. So we wanted to, can we use discovery transcriptomics to reveal these targets or the pathways and then increase the ability to translate data from this system to other systems? So we've been doing this for quite some time. And we do it a little differently than some people. So the idea, again, our protocol, like I described, is up here. So what we want to do, we want to bias our results to early events that precede the toxic outcome. So we know that at 120 hours, where most of the endpoints are detected. So we want to back the truck up and collect gene expression in those exposed animals at the end of organogenesis, where most likely the targets are. We've shown that for a number of compounds. So we want to collect gene expression. So these animals will be phenotypically normal, even though they've been exposed to the chemical for almost 2 days. And development is going to, quote, collapse and reveal those endpoints in 120 hours. So we're not grinding up animals that are phenotypically screwed up, because now you're looking at disease state rather than disease causality, if you want to think in those terms. So we're going to do it at 48 hours, and we've gone through the evolution of using libraries on nitrocellulose membranes to Affymetric arrays to oligo arrays to Illumina, and now we're into other representative sequencing. So we've been doing this approach, this slice of developmental gene expression to chemicals for a number of years. So we have a huge database of what genes are expressed at that time point and what chemicals affect which genes at that time point. So that's becoming even more valuable every experiment. Question is, what concentration? Of course, if you had all the money in the world, you do it across concentration, but of course you can't. So I made this decision years ago that since we do these in pools, small pools, like 6 to 10 animals per pool, and there's no phenotypes. So how do you know that the animals are going to be affected? So what you do is we do our screening, identify the hits, and then we swing back again, grab the chemical again, repeat the exposures in order to calculate the concentration where at 80% of the animals at 120 hours will have the phenotype, which is really important because say if you sample gene expression down here in a pool, your signal is like non-existent. Your signal to noise would be very poor. And then if you're at a concentration way off to the right, you may be at a concentration that's irrelevant to the toxicity. So we want to define the curve and kind of the point we selected when 80% of the animals will be affected. So you should have a really robust transcriptomic signature. So that's how we do all of our phenotypic anchored transcriptomics. So for the pHs, we grabbed a couple members of each one of these categories, and we just use unbiased full transcriptome. And again, for these studies, we grind up the entire animal, and that's actually, we do single cell stuff. There's an advantage. Single cell sequencing is beyond impractical at the scale we're working at. And there's so many problems with the fact that it's a great tool. I love it. It's really fascinating. It's fun. How often is it replicated? there's N of 1, and they say, no, we're sampling 50. No, you're sampling one animal and you're looking at gene expression in 50,000 cells. And you might miss it. Here you can't. If you grind up the whole animal, all the cell types, you don't know where your target cells are. And if you sequence deep enough, you will see the gene expression change. Magnitude might be diluted a bit, so this is the best way to in my opinion, to find transcriptomic responses. We grabbed 2 members from each one. Chemicals, not important. And to our surprise, we were able to group these generally based on the transcriptomic profiles to cluster B and cluster A, which are more similar to each other. So this is an example in cluster B. So what you see in the middle is, and these are the chemicals in that cluster. And all of these gene expression changes in the middle are common. So these things are initiating actually through the H receptor pathway. We've shown it with our knockout lines. These are a battery of genes that are absolutely always turned on together in this system. They may not all be happening in the same cell. That's interesting. When you do pathway analysis, when you assume the pathways in the same cell, but you have multiple cells, that's an interesting, fun journey. But what's interesting is each one of these pHs, in addition to have these common ones, they have a whole bunch that are unique to themselves. This is the 3rd study that we've demonstrated this. We'll take a pH that hits the H receptor. All the toxicity is 100% H receptor dependent. The gene expressions induced by this pH, there's a uniqueness downstream genes for this pH that are different than this one. That's going to be important in other systems, including humans. So it's not enough just to say we detected AHR activation. You really need to understand, are there other pHs, and we've already shown this, there's other pHs that share these other common ones. So I think there's multiple ways that pHs can induce complex biological systems. This is just some of the new biomarker genes that we detected in all of this cluster B ones. So most of them are the same. The rank order is slightly different. So now we have a whole panel of biomarkers that we don't have to do transcriptomics anymore. We can just screen for these and we know which pH we're dealing with. Okay, now diving a little bit deeper. So one of those pHs that we studied is routine. And so Kim Anderson, again, on the Superfund, she does a lot of passive sampling deployment. So she measures it on wristbands on people. She measures it in environmental passive sampling. And this particular pH is found at really high concentrations. It's one of the most dominant pHs in the environment. We live in the Pacific Northwest. We have forest fires every season now, it seems, and the routine concentrations in the air are really, really high. So we want to understand this from a point of view of the mechanism. So this is the structure. It's a substitute. It's an alkylated pH. 2 different groups there. So what happens in zebrafish? So if we run this thing in our high throughput screening, and these are the endpoints that we screen. It's not that important. You see this concentration response in the wild type fish. You do this in our HR2 knockout line that we developed, and if they're largely non-responsive, a little bit of this activity is from CYP1 or HR2B, HR1B, sorry. So there is some activity, but this is an HR2 dependent. This is what routine looks like. Again, characteristic signs of dioxin toxicity, it's routine. And then doing this in a knockout, they're completely normal. So this is HR2 dependent. So these are common endpoints, very specific mode of action. H receptor, 1 base pair substitution was necessary to make this fish non-responsive to this chemical. So boring endpoints, specific cause. So when you start getting all these transcriptomic responses, there's a huge interest right now in the EPA and in clinical space as well, is using transcriptomics to make risk decisions. Not looking at endpoint effects, but looking at gene expression changes, kind of classic biomarker stuff. So is transcriptional adversity a feasible approach to apply to risk assessment? Can we use this model? So can we use benchmark dose or concentration points of departure to identify biomarkers that are causally related to the endpoint that we measured? So what we did, so I had Lindsay Wilson, my graduate student. We wasted a lot of money, but it was important to do so. So we wanted to do a concentration response curve. So we knew this curve, this routine concentration response curve. So at these concentrations, there's actually a phenotype, and at these concentrations, there isn't a phenotype. So we wanted to know, And we knew that the benchmark concentration down here is 5.5 micromolars, where you start detecting that rise. We wanted to know in an unbiased way what gene expression changes at 48 hours were happening before, where a phenotype was going to develop, versus which gene expression changes would be occurring down at the lower concentration. And could we learn anything by doing that modeling? And it's really interesting. So kind of a beautiful transcriptomic response curve. So again, from this really low concentration up to 50 micromolar, the number of differentially expressed genes generally goes up over time. And then so you get more gene expression changes. But in addition to that, so CYP1A I labeled here in red, it was detected really low concentrations, right? But there's no phenotype. So you can think an adaptive response. So two things are going on. You're assembling more gene expression perturbations across concentration, but also the magnitude of the ones that were detected down here go up. So is the toxicity driven by the magnitude increase, or because you've accumulated more gene expression changes? That's a hard question to answer right now. So you can see there's overlap of non-phenotypic gene expression changes and then the phenotypic ones. So it's likely the magic genes are in here. The genes that are causing the effect are in this group, and we're working on that. So when you do this, the EPA is using these benchmark modeling right now to deconvolve gene expression data, and we did the same thing. So if you actually look at each one of these genes that we identified in that cluster, you can see that many of these, their gene expression goes up much earlier before the phenotypic response, so less than a micromolar, and it's really a concentration dependent response. So we can now pretty accurately, with a whole set of genes, identify which pathways are being activated and at how sensitive the system is to those toxicants. All right, now you have the bigger problem, right? Everyone's jumping into transcriptomics and proteomics, and so when you measure these things in an unbiased way, the things which you measure, in this case, gene expression, hopefully the ones I'm most interested are the gene expression changes that are necessary or essential, causally related to the endpoint. Those are mechanistic. Or you might be measuring gene expression changes that are adaptive, right? That's important, but less fun. Or they just happen, right? You have integrated biological systems and you modulate a pathway and gene expression networks are coordinated. They come up, they go down, but they're not related to anything that you're measuring. Can we use systems in vivo systems to kind of sort these out? So we do a lot of gene editing. So this is a kind of important one the first one that we did, where we try to knock out CYP1A. So this was done in rodents in the 90s and the late 80s and the early 90s. When you induce CYP1A with dioxin-like compounds, is that CYP induction necessary to produce oxidative stress to produce the outcome? When I was a postdoc, I didn't believe that mode of action. I still don't believe that mode of action. So this kind of demonstrates I was right. Let's put it that way. So we created this knockout line where we use our guide RNA, and we created, you never know what you're going to get. We have a 2 base pair deletion. So this is stop codon gets put in here. And then we actually had one where it's a 5 amino acid insertion, which was interesting. And we said, what happens when you expose to certain pHs and then dioxin when CYP1A cannot be functionally induced? And so for dioxin, this kind of makes my point. So if you do in the wild type animals, it's very toxic. I mean, TCD is very toxic. If you have either one of these functional knockout lines, we demonstrated that they're functional null using enzymatic activity. There's really no change. So dioxin is still toxic even if CYP can't be induced, right? So CYP1A has absolutely nothing to do in this system with the toxicity of TCD because it can't be metabolized. Routine, very different story. So routine, this is the wild type fish. This is the concentration response curve. If you knock out CYP, it's more sensitive. So this suggests that CYP is really important for detoxifying a routine, which is important. There's even more complicated than that. So we can now use these knockout lines to, I showed you the HR knockout lines. We can determine whether these ligands are HR dependent. We can also demonstrate the role of CYP1A, and we also have the CYP1B1 line as well. And then we had a surprise, right? So you throw this routine and zebrafish, we get this, it knocks these fish out. It looks just like dioxin. The expression pattern is exactly like dioxin. And then we went with the Indigo Biosciences. Jack Vanderhaven's company. And so basically they're using these zebra fibroblast cells, and they have a reporter line that generally looks like this. So we have the H responsive elements driving luciferase activity. And then we threw a bunch of these different pHs. So routine is the most interesting one down here. From very low to high concentration, routine is dead in this assay. Routine does not activate the H receptor in this system. It's like, well, is that just a weird zebrafish thing? So then we went to the human cell equivalent, same thing. So routine is absolutely not a direct HR ligand. So it suggests that routine is metabolized in vivo to become a ligand, binds H receptor, induces CYP, CYP detoxifies it. So we're working our way through that. Trying to understand what the active metabolites are is really challenging, but we're working on that. So now another example, a downstream gene. We have hundreds of downstream genes. So how are we going to prioritize which ones might be essential? So we kept tripping over this gene that we called Wi-Fi kickin' 1. And it kept popping up in our gene expression analysis. And when we did this pathway cluster network analysis, it would always show up near CYP1A, suggesting that it's co-regulated with CYP1A and other genes. So what is it? So first we ran a bunch of different pHs, and wherever we see the induction of CYP1A, this Wi-Fi kickin' gene was right along with it. If CYP1A is not induced, nor the Wi-Fi kickin', the induction is H receptor 2 dependent. The HR nullfish, there's no induction at all. So this is the HR dependent target gene that kind of behaving similarly to CYP1A. But again, what does it do? So it's kind of the fun part when you're doing these things in these systems approaches. You find a new gene, and now you have to figure out what the heck it does. So this is a protein that's never been studied in this context before. It's a multi-protein structure. It's got a bunch of predicted protease inhibitor sites on it, which is interesting. Ligand binding sites. There's some rodent and in vivo studies. Some ortholog can bind differentiation factors and probably modulate their activity. Some evidence for myostatin inhibition. And again, it had never been studied in the context of HR biology. But just look at how complex these types of mechanisms, right? If you're thinking of AOP, so you have a receptor activating HR, chemical activating HR, turns on a whole bunch of genes. Now, each one of those gene products could have other functions, and this one might be affecting the protein stability of hundreds and hundreds of proteins. So we knocked this thing out as well, and we created this mostly gone protein. So most of the subunits are gone, and we said, let's just throw it into zebrafish and use phenotypic screening, proteomics, and gene expression analysis. First thing we found is just in the absence of any ligands, we see that this is this larval fold motor response assay. We see this hypoactivity, so they're less active in the dark. So this is very consistent. And this is way too much to go over, but what we found is just even in the in non-chemical exposure, massive transcriptional effect by knocking this gene out. I mean, like 1,600 gene expression changes. And certainly there's a consequence when you mutate in the mutation with dioxin versus DMSO, it's even a larger number. So this has a profound effect on the transcriptome and the proteome, which I'm not showing here today, but the phenotypes were pretty modest. And this gene on itself doesn't predict hardly, if any, of the phenotypes we see with the pH exposure. So trying to do this for like all the downstream genes for every toxicant gets crazy fast. So instead, what we're doing is we're from pushing lots of chemicals together, we're trying to prioritize which pHs we're going to study and then find the common disrupted gene expression and focus on those and using these knockout approaches. So, all right, I'm going to turn our attention to a different example. So PFASs, forever chemicals. I know you guys are studying these here as well. They're very ubiquitous. They're detected in every biota across the globe. But I think there is a huge challenge still with understanding really the link between the PFASs and human exposure. I think the analytical chemists have been way out front of this for maybe 20 years. And some of the health effects are tied to specific PFASs is not very clear yet. So we wanted to do a similar approach. We wanted to get as many of these PFAS as we can and put them in the same system. The challenge when you do cross-species comparisons, you have all kinds of challenges. You have differences in exposure, you have differences in genetics, and then you're trying to walk yourself with the results from one system to another and try to make sense of it. We think there's value of doing all of it in one system, so at least the only variable is chemical, and that's what we're trying to do. So we got our hands on as many of these. I'm going to talk about the collaboration with the US EPA right now. So the idea is, can we get all these chemicals and then get concentration response curves and identify features that are linked to activity? Because my hypothesis, and not a wild one, is that there's not a chance in the world that all of these compounds behave the same way in the system, even though there's a lot of desire to group them together. So we ran 139 of these in our system, and I'm only going to show it on this one slide. So we looked at, again, the photomotor response assays, and then we calculated benchmark doses, because that's what the EPA wanted to do. And of these compounds, there's like 50 that were active, either in causing the morphological effect or the larval or embryonic photomotor response. So the vast majority, we went from a low to a high concentration. We set our limit at 100 micromolar. 2/3 of them were not active. Okay, and there's some that were, and then some that were active specifically only affected behavior, and some were really specific to morphological thing. So we have now the basis to prioritize which PFASs we should study, and we're sharing that data with the EPA. So one example to really put a nail in it, we took these, I had Yvonne run this study. We picked 4 PFASs from the library that had the same carbon, 4 carbon fluorine length. The only thing that varied by really is the head group that I highlighted here. So if all PFASs behave the same way, we should get the same result with these. We went from 1 to 100 micromolar. We did transcriptomics, and we also did internal dosimetry measures. And right out of the gate, what we found is these 3 at the top, across that 0 to 100 micromolar, we saw absolutely no transcriptomic responses. We saw no morphological effects. We saw some subtle behavioral effects for some of these. But for this one, FBSA with this really nice concentration response curve, very reproducible. So just by the phenotypic screening, these 4 are behaving differently. So and the reason we picked this, there's a dominant view in the environmental tox world that the carbon length is predictive to toxicity. It might be related to bioavailability, but certainly not toxicity. So then we did the anchored transcriptomics like I described, and we could only calculate this EC80 value for the FBSA, which is 47 micromolar. So we said, well, why don't we just run the other 3 at the same concentration and compare them? So maybe they're producing effects that we just can't see. We always, again, since we're collecting these animals at 48 hours, we have extra plates to make sure the response would have happened. They're called holdback plates. And not surprisingly, probably, so the FBSA, almost 2,000 gene expression changes that we see that are, again, they're phenotypically normal at 48 hours, but there's already 2,000 gene expression changes happening. The other ones, we bathed them in these PFASs and we saw absolutely nothing. So it really confirms what we keep seeing, that if you don't see a phenotype for these types of exposures, you don't see a gene expression change. So what are some of these genes? This was kind of interesting. These are developmental exposures, and the pathways that are disrupted are fatty acid biosynthesis, fatty acid. So a lot of fatty acid metabolism. This is what they're actually seeing in humans and in rodents. So these disrupted activities in early development are probably predictive of later outcomes. And then some other ion channels as well are affected. So the simplest explanation is, well, maybe only one of those compounds got in, right? And that explains it. And it could be partially to explain these 2. So for example, you're looking at the number of gene expression changes. And again, this is the only one that produces an adverse effect. But if you look at the body burden, how much got in? So 11 nanograms per mg for this one, but the PFBS is statistically the same. So just as much of this compound got into the zebrafish as this compound. This one did nothing. And this one gave all kinds of phenotypes and lots of gene expression changes. That's got to be important structurally. And so we're trying to use this to understand how these PFASs might be behaving. And then finally, can we find safer alternatives for these really important, commercially important chemicals. There's so many spaces where that are useful, that are not toxic. So we're trying to help in that space. Real quick, we did a study with the National Toxicology Program when they were kind of testing out these various systems. So they created this library. This is just illustrative of the challenges of in vitro screening. 87 compounds for developmental neurotox, and they took drugs, flame retardants, industrial chemicals, pHs, pesticide, and then a small number of negative controls. This is the library they gave us to, we screened it blind. And 27 of these, or 28 of these compounds, they had in vivo data to demonstrate DNT, developmental neurotoxicity. And then other ones that they said were negative, right? So we ran them through our system, and 86 of the 87 chemicals were hit in our assay, some aspect of what I described. And only one of them was a true negative. It doesn't mean that the system is overly sensitive. It just means the system sensitive. So they gave us chemicals that were negative for DNT, but they could have had toxicity in other endpoints that we also detect. So just kind of show the breakdown. We had chemicals that were only affected in morphology and then et cetera. So the LPR is where we really get our sensitivity. It's a very sensitive endpoint. So just so what we did is we went to the ToxCast, the database, and screened for all 86 of these compounds. And what you're looking at on the bottom, I know you don't have to read it, but there's 1,365 potential assays, right, that you could run for every single chemical. And some of these chemicals, like this one up here, has been run through almost all of them. And even then, so some of these assays have a specific mode of action, and that chemical is negative, 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 but in all of these assays, it's positive. We have examples where, this chemical wasn't run very many assays, but most of them is negative, but there's still some hits in some specific assays. So if you wanted to profile the potential of biological activity with some level of confidence for any chemical, you'd run it through all 1,365 assays. It probably cost you $6 million, right? And it might take you a year and a half. We ran it in 5 days, and we could profile this chemical. We can't tell you with any specificity of its mode of action, but we can say this is an active and inactive, and maybe you could apply these other mechanisms. So the in vivo assay is able, it wraps all of these assays together. And that's what we're trying to convey to the EPA. That's still a valuable way of going. The last thing I'll show, so we're pretty good at high throughput screening. We're doing all kinds of fun stuff in that space, but I still think there's a big problem of trying to understand the developmental exposures that are producing these adult effects that are of concern. So how can we do this? So we built a room and built a bunch of instruments the idea is we want to do developmental exposures like we've been doing, and then back the concentrations off to a concentration where we see no developmental effects, right? So sub developmental effects. Remove the chemical and raise them in chemical-free water until sexual maturity, which is around 90 days in our system. And then we're going to run a bunch of adult assays. So we want a phenotype. We're not doing these with a hypothesis in mind, which I think is what the field needs. Do any of these chemicals produce these effects? And then that'll prioritize which ones we focus on. We grow up enough of these guys that we can separate the males and females. That's why we had to wait till they're old enough. And then we run them through systems where we can do auto respiration, fitness, treadmills for fish, basically, shuttle box where we can look at learning and memory in adult fish, shoaling, so social behavior. We can do this in pretty high throughput, response to predator, and this kind of an anxiety test that we do startle response. Let me show you real quick what these instruments look like. This is the treadmill for fish that we can change the speed of the current, and the fish are exercising, and as they're exercising, this really sensitive oxygen probe down here is measuring the consumption of oxygen. And we have demonstrated, this is an example with pHs, developmental exposure to pHs. They have to work a heck of a lot harder to achieve the same level of exercise. So probably mitochondrial dysfunction so we can detect this in a pretty rapid way. This is one of our adult behavior assays. So basically we have a whole panel of tanks, which would be on like a wall, like a 96 wall plate on the wall. Tanks are like this big, and the camera would be facing it. And what the software can do is it can track, it can put a number, you can almost see it. Play. Yeah. It puts a number on each one of these fish, and it tracks where it is in the tank. It tracks where it went in the tank. It tracks the distance between every other fish and calculates all that data. So if a fish tends to be Antisocial, it tries to get away. And if it's social, they tend to get together. So we use this assay. Another assay, these boxes, there's multiple assays here. So we have a whole room of these things. So there's a camera looking down, and these fish are here on these surfaces here and here. There's actually an LED screen. It's not important for this assay. So the fish cannot see out these three walls. They can only see out this side. So in this assay, we're letting them swim. The camera is detecting them, and then we startle them, and you can see that activity. The software can track that activity, and you can see the plots here. And what happens is you can track the activity. There's so much information. How long did it take them to respond? How much did they respond? How long did they take to habituate over repeated cycles? Really complex behaviors that we can do in a high throughput. And another assay is we can project images on these screens like this one. So this is a predator response. So these fish, each one of these is seeing their own private movie theater, and this is what they see in their movie theater. So these fish, the second that this screen is projecting this predator, this is exactly what they do. They get in they hide to get away from it. So if the fish can't perceive the predator like it should, or if it can't respond to it, we can detect it in a quantitative way using this assay. This is a more sophisticated system. So this associated learning test. So basically what we do is we have a single fish, and they can swim under this divider, and it can freely go back and forth. And we want the fish to associate a color, so the correct side versus the incorrect side, which in this case would be red. And then the software calculate, there's no cameras, it's all infrared beams that we built, so it's a lot smaller data set. So the fish is swimming back and forth, and then right when the test starts, it's on the wrong side, so it'll be red. And they're going to get a little gentle shock until they get to the correct side, and the shock will stop. So the software calculates how long were they on the incorrect side, how long did it take them to make the decision, and how long. So we can do this, and we can do this 30 consecutive trials. So we can get learning curves for every single fish, and we can do 48 of these at a time now. So collecting this type of data is really valuable. Of course, once you have that, you have to ask questions, really hard to answer questions of which one of the chemicals have an epigenetic etiology. So we can do the same exact paradigm, and then we could wait till the F2 generation, which was the true epigenetic generation, and ask whether or not this penetrates to the next generation. And we certainly have benzoate is our first example that has transgenerational effects. And so we're linking again. Last thing, we could then kind of tear out these animals and do spatial trend. We could do all that, but I don't want to do it that way. So instead, what we do is anything that happened here started here. Everything we do is focused on the beginning of an event, not the effect. And that's where I think we can predict chemicals that can initiate these pathways that can predict an epigenetic. We still have to measure the evidence of epigenetics, and we certainly do that with bisulfite sequencing, for example. But that's where I'm going to end with it. I'm going to go up to the beginning. We're pretty disciplined that way. This is again the benzoate pyrene example. We all the way to the F4 generation. This is this embryonic photomotor response assay. We have this hyperactivity response that goes on forever. So we're trying to explain which. We did a full genome bisulfite sequencing, and this is insanely complex. The number of methylation changes from that developmental exposure are impressively large across the genome. All right. Yep, and there were 4 endpoints that were epigenetic, and only 2 of them really held. The other 2 kind of diluted themselves out. So we don't understand the exposures associated with human disease. I want to do that. I want to really crack open more possibilities of not just observing effects, but understand which exposure. The zebrafish, we can identify associating between exposures and outcomes. We do real world mixtures. We can actually tackle mixtures finally. We can discover the targets of these hazards. We can unravel these tox mechanisms by using knockout and transgenics, et cetera. And then I think real importantly, there's all the rage right now. If any of you go to the big meetings, there are new approach methodologies to try to replace the use of animals. A lot of them are not going to be valuable. I'm telling you that right now. Some are going to be really valuable. And I think these in vivo models might help make that decision on which ones are valuable and which ones aren't. I got a whole bunch of folks in the lab. I talked about some of the ones in bold today, and important collaborators at NC State and Katrina Waters, and then certainly EHS for funding. And really, I only showed you some of the instruments that we built over the last several years, but I've had an engineering team in my group for 15 years, and to be able to conceive and then build instruments that are actually useful is really, really fun. With that, I'll take any questions you might have. Thank you. There's three zebrafish, right? So I should go all three when I go stop. And the H receptor 2 is the functional equivalent to the human. So all of our, we have homology model, the combined domain. We have not found a ligand in zebrafish that isn't a ligand for humans or vice versa. So the ligand binding part of it is really well preserved. I won't go as far as to say is that the consequence of those bindings wouldn't necessarily be the same. You know, selective HR modulator here for leading under the night. I think that holds here, so we're working in that space. But the reason I picked the A receptor to start this field was because I thought it would be really functionally conserved, and for the most part, it's there's other pathways that they depart a lot sooner than that. But I'm very confident that most of what we discover in zebrafish along the HR axis is very well contributed.
Amazing, Doc. So I was really interested in how you're calculating your benchmark dose. So I was wondering, can you tell us a little bit more about how the benchmark dose that you find in the zebrafish that the humans over?
Yeah, great question. And it's a really important question because I think it's in vitro studies really problematic because of report nominal water concentration. They are clearly required, right? All cell culture studies are required to talk, etc. So the way the chemical is behaving in that aqueous solution, it's meaningless to just report concentration in the dish. And we do, I think showing today, kind of partition box to predict when you throw the chemical in the dish, how much it is likely to be volatile, how much is likely to bind the plastic, how much will be effort, or metabolites, and all of that is happening in vitro. So when you put in a concentration in or bind serum, you're doing serum. So even equate any in vitro studies to two in vivo studies, you can't use nominal water. So we're trying to model these the total single compartment tissue concentrations for all of our chemicals. If you can't measure for all of them, then no analytical chemist in the planet would want to do that for us. So what we're doing, we're trying to take chemical properties and then try to predict with some level of certainty model the likely partitioning tissue doses, which then it might be able to compare to tissue concentration. So I think the challenge is real. It's a great question. But it's not unique to this system. It's more people need to measure the tissue, the cell concentration in vitro, which would also probably help to translate those results if you use more than that.
So we talked a little bit this morning at breakfast about drug development, and you started with your comment on drug development, that it's easier than toxicology, but there's a flip side in drug development that is actually really, really hard and impacts what I do as a hepatologist, and that's drug-induced liver injury, which is still a huge problem and major expense for drug companies. And so your talk really reminded me of there's really converging evidence from NTP that's on DILI, and so they published several years ago when Dept was there, something called, it's like 3-day or 5-day study. Have you heard of that? Where they would give DILI drugs versus non-DILI drugs, mice for 3 or 5 days, and they've done it in cell culture too. And basically every drug they threw at their system that was associated with human DILI caused a massive change in DEGs. And those that didn't cause DILI, and this change preceded the observable toxicity. So it's very much the same. Yeah, it did, and they said it didn't matter what the specific changes were. It was, and in every drug the changes were actually different. but they were common and that they suggested you could possibly predict or use this as a screening, kill the drug before it got too far along in development. And this reminded me of that. And so it's, to me, converging evidence. So one of the questions that I have for toxicology, is this going to be something that's true to all systems that you're looking at in the body? And is there a threshold change in the, how many DEGs do you have to see to say, this drug or pollutant and over, it's going to be a PFAS replacement is going to be a problem. Is that
I mean, that's the idea, but that's the different goal, right? So like the drug discovery folks are probably more, they would love to identify those earlier, not right. So the EPA, when they first started doing toxicogenomics type studies, and they weren't requiring collection of this data, but they would take it. The fear was that someone might try to make a decision based on a transcriptional change that there is no evidence that it's associated with a true in vivo response. So they were like, wait a minute, that's actually what's holding back implementation. And it's rightly so, because inferring these molecular responses as predictive of a favorable outcome isn't fully established yet. I think we need to do more case studies to demonstrate where it holds, and knowing that sometimes you're going to find one that break those rules. But is that any worse than how we're moving out? But no, I totally, they're like surrogate measures of molecular responses that you can't explain, but it's useful information. A decision can be made in that example, even if they can't explain it yet. So that's one of the page two we're trying to do.
The second part of that question, in your model, is there a minimum number of DEGs that you've seen that's been associated with a phenotypic response?
No, not.
Is it always thousands or hundreds?
No, there's some compounds that have a really specific target, and a small set of gene expression changes are most of the ones that we think we can understand. Others, I mean, PFAS, I think it's a nightmare. PFAS, I personally believe, do not have a specific target. I think every assay is going to detect a different target if you have a response, because of the way these structures interact with proteins. So that's going to be like a QSAR type approach to be challenging everything with PFAS. So I don't think they're all going to be in the same way. And but I think we're going to get there with some of the structural modeling and the interactions that identify which PFAS and what the most probable targets of the systems are. I hope that's where the field ends up. We can't just keep lumping. I mean, drug discovery is really good about using medicinal chemistry to make chemicals do interesting things. Right? Toxicologists tend to say, "Oh, it kind of looks like a PFAS," right? They're all going to be, no, you can't. I mean, what small changes can make a total difference in drugs. Certainly does in environment. Thank you.